Great. Well, thank you so much for the invitation to be here. I realize this is an audience that probably doesn't get too many biophysics talks, but that's good because then I can teach you something, I hope.
Yeah.
As I've mentioned to someone earlier, the fact that I'm in DAMT probably is perfect proof of the rule in Cambridge that what goes on in departments and the names of departments don't have much to do with each other. Because although I am a professor of mathematics, we actually have a working cell biology lab in the basement, which very few people know about, including people who actually work in the same building. So I'm going to tell you today about some work which I think perhaps fits in with the theme of the center, involving a problem in evolutionary biology. And to put it in context, I think the best way to begin is to ask you, as I've asked many audiences, the rhetorical question: if you had to list the most important unsolved problems in science, not with science, but in science, what would you say? And I'm sure in this room you'd all agree the origin of life is probably top of that list, and maybe number 2 is something like the nature of consciousness. But then it gets into arguments about what would be number 3. But at least for many of us, high on that list is the origin of multicellular organisms. That is, how it was and why it was. The simplest organisms that first appeared on Earth and were surely unicellular, akin to a bacterium, evolved eventually to become larger in the sense of having more cells, and also more complex in the sense of having more cell types. Now, if you think about many of the biological processes that occur for an organism in the primordial soup, a lot of physical issues matter, physical processes like buoyancy, diffusion, and mixing, and light propagation. And these are physical problems. And it's not surprising that you'll find lots of physical scientists, as well as biologists, working on this kind of question. So that's what I want to tell you about today. And I'm going to use a particular biological phenomenon called phototaxis, which is steering toward the light, to talk about the nature of evolutionary transitions from single cell organisms to multicellular ones in the context of this kind of control problem. The place I want to begin actually is some very interesting data that was first presented to the world by Bell and Loors years ago, and then was reinterpreted by John Bonner. The late John Bonner was a very distinguished evolutionary biologist at Princeton who spent basically his entire life worrying about the evolution of biological complexity. So in an astronomy department or any place dealing with the cosmos, log plots are probably the way to go. So here's a log plot. For many different organisms in the animal and vegetable world, if I can call it that, the total number of cell types in the organism by some method of counting as a function of the total number of cells in the organism. Okay, so a bacterium is 1, down here. It's one cell, and it's one cell type, and that one cell type manages to do all the functions of life. Now humans, like all of us, are about 1/10 of a cubic meter in volume. And among friends, a cell is 10 microns on a side. So that makes for 10 to the 14th cells. And by the method of counting here, we have about 210 distinct cell types, just off the chart. So now you might look at this graph and imagine there's a power law somewhere to be found here. A straight line that would go through all the data, and perhaps
an astronomer would be convinced.
An astronomer would be convinced. Yes, thank you. Yes, it's funny because when I talk to condensed matter physicists, they yell at me and say, "How could you possibly draw a line?" but I'm not sure what you'd learn from the slope. But it's pretty clear that there is a general trend that as organisms get larger, they get more complex in the sense of dividing up processes into specialized cells. So if you are brought up in a world where reductionism is the norm, then it's kind of obvious that, assuming my thing works, that we should look in the lower left corner here, if we want to make some progress on this, and try to study, as a function of increasing cell number, the first appearance of 2 or 3 cell types, and see if we can make some progress in understanding what's going on there. And I submit that if we can't do that, it's unlikely we're going to be able to understand the whole thing. So I'm going to tell you about a research program that we started many, many years ago, which is essentially to use certain classes of model organisms to start addressing this question. What are the first steps toward multicellularity, the first steps toward division of labor, from the point of view of physics and math, as well as biology? Now, in any of these problems in biology or biophysics, it's very important to adopt the right kind of, quote, model organisms. So a model organism in biology means an organism which the community has decided collectively we shall study one reason or another. Usually it's easily grown, it's easily genetically engineered, et cetera, et cetera. But if we all study it from many different perspectives, rather than studying 53 different organisms, each from our own one perspective, we'll get a better handle on what's going on. So the question is, what is the right set of model organisms for this kind of problem? And the answer was provided by Antony van Leeuwenhoek. How many of you know Antony van Leeuwenhoek was? Good, one. Right. So he wasn't the inventor, but he was sort of the perfecter of the early microscope. And his microscope, which you can go to the Boerhaave Museum in Leiden, the Netherlands, and get a copy of, as I have. You were just there?
We were just there. We can start with
yes, there are all these lenses by Huygens that are there also. And right near them is this. So notice the size. It's sort of like a large postage stamp. Brass panel with an XYZ translation stage, ending in a pin, sitting behind a single millimetric lens of exquisite precision. And it was figuring out how to make those lenses that revolutionized microscopy at the time. And with this amazing microscope, where you just put something there and hold it up to the light in your eye, van Leeuwenhoek saw many, many things that revolutionized biology. And one of the things that he saw, he described in a very famous letter in the year 1700 to the Royal Society. And you can actually go to the Royal Society, see this letter. And it has a title which I wish I could use in a paper, part of a letter from van Leeuwenhoek concerning the worms in sheep livers, gnats, and animalcula in the excrements of frogs. So in his wonderful English, he describes basically looking through the microscope and discovering an organism which he drew by hand in his figure 5. It's about 0.5 millimeter across. It's covered with green dots. It has larger green spheres on the inside, and it perpetually spins and moves about. And it was not until almost 60 years later that it was given a name by Linnaeus, the namer of everything originally in biology, in his great book on taxonomy. And the very last entry in the entire book is that of Volvox, the name given to this organism, from the Latin root volvere, which means to roll or spin. Yes?
That thing is, there's a pin. And when you can't put a drop of water on the end of the pin, I'm just wondering what.
You can put a small drop of water on the
Okay, so it was
yes.
A small drop. Okay.
Yes, I have it in my office. We can go play. Let's see. It actually works. It's amazing. In fact, many people doubted that he could see what he claimed to see. And Brian Ford, who's a great microscopist in the UK, took his original microscope, which the Royal Society has, put a CCD camera behind it, took pictures and showed that he could do it. Yeah. So it was given its name because if you read the Latin, the organism appears to spin around without pause. Now, not P-A-U-S, but P-A-W-S, not pause like animal pause, because at the resolution he could see this thing spun around for no obvious reason, but it actually has flagella that cover its surface, that beat in a very characteristic manner, that spin it and move it through the fluid environment, and I'll tell you more about that. So here's, reluctant to switch. There we go. I hope that works. Here is a modern movie of Volvox, which made a cameo appearance in the BBC documentary, The River Tay. So this is a real-life, real-time movie of Volvox organisms. They're about 0.5 millimeter across. Each of the organisms has about 1,000 cells on its surface and a smaller number of daughter colonies that grow up on the inside. And as it swims, it spins because the flagella that beat, that are mounted 2 at a time on each of those external cells, beat basically from the North Pole to the South Pole, but with a slight east-west twist. So the whole thing spins as it swims. And this spinning and swimming is very characteristic of a huge number of microorganisms in the world. And it's part of the story of how they sense light and move toward light. So it's mesmerizing to watch. I mean, you can come to my lab and watch it. It's really quite striking. But I want to point out a couple of important things about this organism. It's multicellular. It has maybe 1,000 plus 12 cells on the inside, which is the reproductive cell. But it has no brain. It has no neuronal connections, no central nervous system between the cells. They're basically, to a first approximation, just independent players mounted in a polymeric goo. And yet, it will turn out to be capable of doing amazing steering through the environment and other processes. Okay, so, hello. Now, nature's been very kind to us in the sense that we're presented with Volvox as an interesting organism, but it's actually the sort of endpoint of a whole lineage of green algae that were given by God to us to study as mathematicians, because the fundamental building block of Volvox are the individual cells on its surface, of which there might be 1,000. And those individual cells are essentially identical to a single cell organism called Chlamydomonas, which is 10 microns across. Remember, your hair, if you have any, is about 100 microns across. So it's 1/10 the size of the hair. And Chlamydomonas has 2 of these flagella that it uses with the breaststroke to swim through its fluid environment. And there is a series of species. These are not transient accumulations by adhesion. These are distinct species that consist of 2 to the n Chlamydomonas-like cells arranged in some geometric pattern, which originally for small number is basically a plate, but eventually becomes spherical and can reach species that have 50,000 cells and can be several millimeters across, starting at 10 microns. So what you see here is that we have a whole lineage of organisms spanning a huge range in cell number and a pretty large range in size, 3 orders of magnitude, with which to try to understand scaling laws for the functioning of them as collective objects. But interestingly, all of these species to the left of this line have the feature that every cell does every function of life, meaning reproduction and vegetative aspects. Whereas to the right of this line with Volvox proper, there is a differentiation between reproductive cells that live on the inside and sterile so-called somatic cells that live only on the outside. They're like the galley slaves that just do the swimming. So there's a fundamental, what's called germ soma, germ reproductive cells, soma somatic, differentiation that sets in at a particular size, particular number of cells, particular radius. And I really would like to understand what determines that size. Why does it suddenly become evolutionarily advantageous to have 2 cell types there? And if we can't answer that question, I don't think we're going to get to that power law.
Are there non-model cells that overlap this, like things that are as big or bigger than Volvox that are just one kind, or things that differentiate that are smaller than Volvox?
So actually, no. What really happens is that it's a slightly fuzzy line,
Okay.
because I kind of simplified it, but some species like here have a partial separation of the cells on the outside in terms of who does reproduction and who does the other, but it's not terminal differentiation like you have here. So basically, the ones on the inside grow up from specialized cells that only do reproduction. The ones on the outside never do reproduction. And there's an interesting biochemistry and genetics about how that happens. Yeah. So there are obvious questions. You know, what are the driving forces for the transition to multicellularity? And how does some kind of collective behavior that allows all these apparently non-communicating cells to function together as an object emerge? What are the rules? Okay, so the previous one said, you know, those were model organisms for evolutionary transitions to multicellularity. I just want to point out over the last sort of 15 years or so, we've been able to see that these organisms are also model organisms for many problems in biological fluid mechanics, separate from what I was describing. So in addition to the general issues I just outlined, the flagella with which these organisms move, which are conceptually and proteinaciously identical to the cilia in your respiratory system or in the back of your brain and elsewhere, synchronize with each other as they beat. And there's a lot of interesting fluid mechanics and stochastic processes involved in that synchronization process. And that's been one of the things that we've been able to figure out, we as a community, on the basis of using these organisms. There are other interesting things that happen where the organisms interact with each other through their hydrodynamic flow fields and can actually form interesting things called hydrodynamic bound states, which was just a random discovery that came out of watching these things. The Phototaxis I'll tell you about, but also because the organisms are large, easy to grow in the lab and easy to study under the microscope, you can measure the flow fields around them with incredible precision, because basically they're very steady and you can average and average and average. And you can get beautiful streamlines and mappings of fluid velocities away from the center and connect the dynamics of those thousands of flagella on the surface to the large scale flow in a way that's not possible with other kinds of organisms. And then there are some interesting physicsy problems having to do with the dynamics of tracer particles in the medium that they stir up that are also amenable to study with these organisms. Okay, so just to convince you of what I said in the beginning, we actually have a cell biology lab in DAMP. And if you go downstairs, you'll see we have these large refrigerator-sized objects called diurnal growth chambers, which are basically temperature-controlled light boxes where there's a day-night cycle of temperature and of light. Because these are photosynthetic organisms, they must have a light cycle just like the plants all around us. Right, so we grow them just as biologists would in Erlenmeyer flasks with air bubbling to have them synchronized and ready for the experiments of the day. The way we study these things under the microscope is as follows. We use very simple microscopes, but basically we use technology from the world of in vitro fertilization or electrophysiology. So we could hold the individual organisms on micromanipulators that have micron scale precision. So we can basically aspirate a tiny organism at the end of a glass capillary and hold it right above a microscope objective. And with that, so this works, we can put it in a fluid medium in a specialized chamber. And if I do this, and you look closely, you see that we go from the scale of this glass capillary, which is about 1 millimeter or 2 across, all the way down to its tip, which might be neck down to something like a few microns. And with that, then we can hold organisms that small and try to study how they respond to light or anything else. And so what I'm going to tell you about now with this preamble over is a story about how we've tried to understand the sort of underlying dynamics of steering organisms that don't have a brain as they go from a single cell organism like Chlamydomonas all the way up to Volvox, and using an intermediate organism known as Gonium, which consists of 16 cells in a plate-like arrangement. And this allows me to use one of my favorite words, a triptych of organisms. It's probably the only time I've ever used it in science for the study of phototaxis. So notice this is about 10 microns across. This is 16 cells up to maybe 40 microns, and this thing can be 0.5 millimeter, close to 1 millimeter. So it's quite a range of sizes and 3 orders of magnitude in the cell number.
And on the Gonium, there are flagella projecting out.
Yes, I'll tell you more about the structure in a moment, but it is a very interesting missing link in a sense between these two. This organism swims with a breaststroke fashion like this, primarily. This one has peripheral cells where instead of the flagella beating in opposite orientation, they beat together, but it's organized in a kind of spiral way, so the whole thing spins. But the central 4 beat like this, so it moves forward. So it's kind of figured out how to accomplish both tasks. And then by the time you get here, it's a sphere, and everybody's roughly doing the same thing, and then it spins and rotates. So all of them spin, actually. This one, although the 2 flagella are almost identical, but not perfectly identical, and they beat slightly out of the plane. So it spins as it swims, this spins as it swims, and this spins as it swims, each with unique frequencies, characteristic scales, etc. And so that will help us tie together our understanding of this elementary process called phototaxis.
Is spinning important?
It's super important. You will see. Yes. But in fact, if you look back in the old literature, which I'm very fond of, you will see that as long ago as 1901, and probably well before, people in the biology community were commenting on spinning motion, which you just see everywhere. And Jennings wrote this amazing paper. He was not a mathematician, as far as I can tell. He was a biologist. There's no math in here at all, but he figured out just by observation a very important point, which is not obvious. And it goes like this. He said, when I see an organism moving along a helical path, which many of them do, so they're rotating around a body fixed axis, but they're also moving on a trajectory along a helix. He said, the body of the organism bears a constant relation to the axis of the spiral. That is, the same side of the organism is always directed toward the outside of the spiral. And it's not for the same reasons that we see the same side of the moon. Right? It's something else, right? So somehow, whatever propulsive mechanism they have, some body part that you pick, I don't know, this flagellum or that flagellum, always is on the outside, or another choice would be always on the inside of that helix. And I don't know what I do. It's not obvious to me that if I actuate some body parts in some sequence that I should be like that. You know, why doesn't it spin in some different way? But he understood that this was the case. And he made an argument that this device revolving around like that might sort of compensate for any imperfections in the actuation mechanism. But he also had a very interesting observation. There's an organism called rotifers, which have the ability to actuate one or several of these flagella. And when they're near a surface and they swim around, they tend to swim in circles. But if they manage to lift themselves off the surface and actuate a new mode of motion, then they form a helix. And so what you can infer from this is that if a body has actuation that rotates around two different axes at the same time, like Chlamydomonas spins this way, but if it also has a differential forcing from the two flagella, it will spin around a different axis. If it actuates around two axes separately, then it produces a helix. It's a nice math tripos problem. So he understood all of that in 1901. Okay, so let me tell you some other features about the way what's called phototaxis works in these organisms, that is motion toward the light. If you look down the anterior pole, the top part of Volvox, you will see on every cell a red spot. And that red spot is known as the eye spot. It is the locus of a primitive photosensor that is common to all these organisms, by which it senses light and adjusts the beating of these flagella accordingly. And the way that eye spot works is illustrated here from a recent paper that was actually studying an interesting mutant of the problem, but it illustrates the main point. So the eye spot consists of a red protein layer that is what you see, but the photosensor itself is actually on the outside of that, closer to the cell wall. And so the two flagella of the organism are known as cis and trans relative to the eye spot. Cis is closer, trans is further. And when light falls on the wild type organism, it's eye spot, this protein layer turns out to act a little bit like a quarter wave plate in optics. It reflects the light back, thereby giving an extra strong signal to the photosensor. And that signal is turned into chemomechanical activity in the cell that modulates the beating of the two flagella. In particular, it makes the cis flagellum beat stronger than it was previously. Actually, the trans one beats a little less. And the net effect in this case is that the organism turns away from the light. So the typical organism is actually negatively phototactic. It tries to hide from the light. Okay, but if it were the other way around and it wanted to go toward the light, then this one would be the dominant one. But this protein layer has the feature that if the light is coming from behind the cell, it's blocked from the photoreceptor and the cell doesn't see it. So it only senses light from the forward hemisphere. If by forward, I mean the outward normal to the cell. And that directionality is very important. And we know it's important because there's this amazing mutant that's missing the eye spot. So it's got the photosensor, but it's missing this protein layer. And then you might think, well, it's going to get pretty much the same signal, whether light comes from the front or the back, so it won't know what to do. But amazingly, it turns out it does know what to do because the cell body acts as a lens. And light coming from behind is of greater intensity than the light coming from the front. And so it actually goes the opposite way you'd think. Well, actually it goes in a direction at all is interesting, and it's the opposite from what you think. So algal optics are really interesting, and the directionality is fundamental to the problem. Okay, so the way we're going to study this is to not necessarily track the organisms as they're spinning through the fluid, because that's hard to do to understand what the flagella are doing, but we're going to hold them on micropipettes, as you see here, like you see with in vitro fertilization. You've seen pictures of egg cells with sperm being injected. And we can subject them to light, which we bring up with a fiber optic light guide, that we can turn on and off at whatever frequency we wish to mimic what happens when the cell is spinning. Because if the cell axis is this way and it's spinning and the light is over here, then the photosensor is in and out of the light and it gets a periodic signal. And somehow the ability to swim toward the light is getting rid of that periodic signal. So it's a very interesting kind of control theory problem. Okay, and there's an important separation in timescale here that matters when it comes to developing a theory, but also even just in analyzing data. So the flagella of these organisms beat at frequencies anywhere from 25 to about 50 hertz. Okay, so in milliseconds, we're talking about periods of beat of maybe 20 milliseconds or something like that. The time it takes the organism to spin around its body fixed axis is some much larger fraction of a second, probably 10 times larger. And the time it takes to execute a phototurn, to actually turn toward the light, is even longer by a factor of 10. So that makes it possible to sort of coarse grain this problem on several different timescales and get a sensible theory. And we have these two kinds of experiments. We hold individual cells, as I showed you, with an LED we can bring up to it and do high-speed imaging, or we can actually track their motion in 3 dimensions in a specialized device where we are free of wall effects and thermal convection and all sorts of that, things like that. And we track them with 2 orthogonal CCD cameras, and so we can build up their full 3D trajectory. Okay, do stop me if you have any questions. All right, so let's start back in time when we first studied this problem, which was more than 10 years ago. We were interested in Volvox. I don't know why we started there. We should have started with Chlamydomonas, but we started with Volvox, but just because it seems so rich and complicated, I suppose. And so here's the situation. You have an organism that spins around a body fixed axis. It sends flow along its periphery. And if you look microscopically, you'll see the individual flagella of one of these somatic cells. And so we'll bring an optical fiber up to it and shine light on it. Now remember, every cell on the surface has one of these photoreceptors, and we just want to see what happens. So it's an old movie, but important. So here's the cell, here's the micropipette. And when I start the movie, you'll see tracer particles to allow you to visualize the flow. So the light is off to begin with. Now watch, so it's moving quickly. When I turn the light on, you see it slows down. And then within a second or two, it speeds up again.
Is this real time?
That was real time, yeah. So there's a very short response, rapid response to the light level going up, which is to slow down the flagella, slow down the fluid flow, but it recovers. And if you do a technique called particle image velocimetry, where you basically track the motion of those particles and get a vector field of fluid motion, you can make a simple graph of the flow speed around some particular section of the cell, of the colony, as a function of time, as we go from light off to on. And what you see in red here, which is the average over many runs, is a very rapid decrease in the flow speed and then a slow recovery. So this is what's known as an adaptive response. It's just like you walk into the bright light outside, your eyes are shocked through a bit, and then you eventually get used to the light.
Is this something with like a 1 second period superimposed on a longer?
I wouldn't read too much into the noise. It's quite random as far as we can tell. Yeah. I mean, you have to be careful when you get too close to the surface, you're seeing the individual dynamics of the flagella as opposed to some coarse grained velocity. Now, when you look at this, depending on your scientific background, you might think this is some very nonlinear process like what in mathematical biology is known as an excitable medium, like action potential in a neuron, where if you stimulate a neuron with a voltage or current from the outside, if you make a small perturbation, you get a very small result. But if you make above a threshold perturbation, you get a huge result. And it's a very nonlinear system. But this is not like that. It's a perfectly linear system where this response is proportional to the signal that you put in. And it turns out, on a purely phenomenological basis, this biphasic response, rapid decrease followed by slow adaptive increase, can be described by a simple 2-variable model, which turns out to be used all over the place in biophysics to describe adaptive responses to chemical signals of all types. And the basic idea is this: let's say that what I am observing is called the photoresponse P. And there will be another variable in the problem called H, meaning hidden, which represents all the biochemistry responsible for the resetting, the adaptation of the process. And so there are 2 time constants, a response time, which is short, an adaptive time, which is long, and a signal. So you have P, signal, H, the hidden variable, and P. And this is basically just 2 capacitors that are talking to each other. Right? So if I start with a situation where there's no signal, and then there's no H and there's no P, 0, 0 is a fixed point here. If I suddenly turn signal on, then the first thing that happens is that on a very short time scale, P tries to equalize itself to the signal. So you get a rise in P. And it's not till much later that H wakes up and says, no, there's a signal. And so I want H to get to equal the signal. And then when H gets closer to the signal, now P is reacting to the difference between these 2. which is getting smaller and smaller, and that gives you this resetting. So it's just two capacitors that talk to each other. And so the picture you could have here is that the fluid speed at any point on the surface is whatever it was before I turned the light on, minus a little bit proportional to this p variable. So that's the first indication that there's a dynamical response with two timescales in the problem. Now you can ask yourself, where do these variables come from in the biochemistry? And the answer is, we still don't really know, but most likely the adaptive variable has to do with calcium influxes into the cell. Now, you can just take the Fourier transform of that 2x2 system and get the frequency response. If I now think of a signal which is from the outside, as I do with my light, turned on and off at some frequency omega. And what you discover is it's a bandpass filter. It will have very little response at low frequencies. It will have very little response at high frequencies, and there'll be a peak in the response at intermediate frequencies. So the two variables, the square root of the product, gives you the characteristic peak frequency. And the amazing thing that happens, and here's the mathematical formulation of that, it's kind of like a Lorentzian, is that I can now ask myself, what is the relationship between this characteristic frequency, these are measurements from our experiments, and the rotational frequency of the organism. And it turns out that if you study a large range of Volvox sizes, and you can get that range by just watching them as they grow up, they enlarge, you discover that their rotational frequency around their body fixed axis is a very strongly decreasing function of their radius. As they grow, at fixed cell number, they slow down. And the only ones that are very good at phototaxis are in this, basically this purple band, which are reasonably large. And that band corresponds to almost exactly the peak frequency here. And so what's going on is very simple. Suppose the light's coming from there, and my body fixed axis is here, and I'm spinning around this way. This little patch of the surface, full of those cells with photoreceptors, rotates into the light. And the first thing that happens is their flagellar beating goes way down, and it's not until about half an orbital period later that it recovers. And if the recovery time is matched to the orbital frequency, the way I described, then the net effect is that the light side will beat less, and the dark side beats stronger, and you go to the light. It's that simple, if there is tuning. If it's mistuned, then you won't have this nice hemispherical separation between the two. So you can further mathematize this in the following way. You can say, a theorem from fluid mechanics says, if I know the velocity across, the slip velocity across a sphere, the radius r, the vector u as a function of polar angles on the sphere, and I do this integral, I integrate over the surface of the cross product of that velocity with the normal vector to the surface, divided by 8 pi r cubed, I can actually calculate the rotational angular frequency of said sphere. This is a theorem due to Stone and Samuel some years ago. And so in this coarse-grained picture where I'm not describing what the flagellar are doing, I'm just talking about the surface velocity of the flow field, I can do that here. And what you can see is that, of course, if the fluid velocity were the same everywhere, this integral would be 0 by symmetry. I must have an asymmetry in order to turn. And with this picture, where the fluid velocity as a vector was what it was before with some correction function p, then I almost have the complete story. I just have to say if the light is coming from somewhere, then the direction of that light evolves by the same solid body rotation omega that I had before. So you can turn this into a little computational model where I'll use blue to represent where on the sphere the beating is strong, and red to represent where it has been brought down transiently by the photoresponse. And what you'll see is that initially it's low here because it's facing the light, but as it turns, everybody recovers their happy beating because they're constantly in the light, and the system will line up with the light. And notice there never had to be any discussion between cells on the surface here. They're each responding to the light according to this rule. And that's the most important lesson I think you can draw from this, which is that in this first step toward multicellularity, it's not necessary to have discussion among the cells that are mounted in this surface, as long as they are responding to the signal with the right program of response, which is presumably inherited by evolution. And I like to say that a good analogy with this is if I went to a church on Sunday morning and I looked in through a window in the front door but didn't disturb anybody, I would occasionally see that everybody stands up, and then everybody sits down, and then later they stand up again. And I might think that they're all talking to each other and saying, no, time to stand up. But all they're doing is following the guy in the front. And that's the same thing that's happening here. They're each responding to the light with a programmed response. And the net effect is we think it's collective behavior involving intelligence, but it's not. Okay. So
In that example, there is communication, right?
the people.
Because I'm watching the person in front of me. There's.
Yes, but you needn't be. You could have just a tunnel vision to the guy in the front and that would have been sufficient.
I see. And in this case, there's no communication.
It turns out it's not quite that there's none.
Okay.
There's hydrodynamic communication between them, which can lead to synchrony among the flagellar beating. But in terms of the photoresponse, there's no known communication.
And all those people are in the room by a priori communication.
True.
Which is, there's that other
Yeah.
problem.
These have no choice because they were born that way. These are stuck that way. This is true.
Yeah, but they got together.
They got together, yeah.
There's another issue, which is getting those guys together.
This is true. This is true. And so that brings us to Gonium. Thank you. Yes. So again, I emphasize these are not transient accumulations of cells by adhesion. These are born this way. And so here's Gonium with 16 cells. This is from the web. It was studied more than 100 years ago in detail, but here's this extracellular matrix that connects everybody together. And when this reproduces, this cell or this cell or this cell would produce a 16 cell daughter. So you'll end up with 16 times 16 all packed together in some strange way, and then they go off. Okay, so here again, as I say, it has this feature that the peripheral cells beat their flagella in the plane, roughly, not quite completely, which leads to spinning, and the central ones lead to out-of-plane motion. So some years ago, more recently than the last one, we thought it was time to study Gonium and see what goes on. So here's how Gonium swims. There's a little plate-like structure that rotates, moves forward, and it actually moves on a helix. Not a very precise helix, but it's clearly a helix. Here are some of the numbers. And if you look in a quasi-two-dimensional chamber and you turn light on and off on either sides of the chamber and speed up the movie by about a factor of 10, you can see they all get phototactically sent this way, and then you turn the light over here and the other way. So, yeah. So they really do respond very well to the light. Yes, remember, it's sped up by a factor of 10. But it's clear they can do phototaxis and they do it well. And how does that work? And is it in any way adaptive as the other one is? So it's actually a little trickier to study something like this, but you can do it. And again, you hold it on a micropipette, as I showed you before. Now you do particle image velocimetry just before the light, just when the light is turned on, and afterwards. And you see if yellow represents higher speed fluid flow, you can see there's an asymmetry between the illuminated side and the dark side, which eventually starts going away. And if you plot the local fluid velocity just normalized to 1 for what it was before as a function of time, you again see a noisy but clear biphasic response. And you do this for hundreds of colonies, and you get a cluster of what the adaptive and response timescales are. Here's the line of equality, and everything tends to be above that, meaning that the adaptive time is significantly longer than the recovery time. So it's pretty much the same dynamics as in Volvox.
A handful of them are on the line.
Yeah, that's true. And I think I'll get to this point later, that it makes sense that there's typically a strong separation, but it's not necessary.
Okay.
And there is a really interesting issue, as people always say, it's biology, so it's noisy. I don't have to say that, it's cosmology, so it's noisy. But it's biology and it's noisy. And actually what one would like to understand is how robust is the tuning mechanism to changes in these time constants. And I'll get to that at the end. Okay, so the last couple of years, we've spent a lot of time trying to think about how would we adapt, speaking of adaptation, how would we adapt this kind of picture to Chlamydomonas if it's appropriate? And now Chlamydomonas is really, it's the most elementary of the organisms, but it's also particularly complicated what it does. And it's illustrated here in this diagram. So let's imagine the light is coming from the right as shown by these arrows. And remember, I have a cis and a trans flagellum relative to the eye spot. So the organism is spinning at about 2 hertz around this central axis. So at any instant of time, imagine that the cis flagellum is facing the light and the eye spot sees it. Then that's, in this positively phototactic case, then it turns out that the cis flagellum will slow down its beating, change its amplitude actually, and the trans flagellum will increase. So transiently there's a turn toward the light, and that makes sense. However, half a turn later, the two flagella have switched as to which one is facing the light. If you had the same response, you'd go away from the light. So it has to be the opposite response, that now the transflagellum, well, actually, whichever one I said before, the cisflagellum must go up and the transflagellum must go down. So in some sense, it has to forget whatever it did here by half an orbital period later to do the opposite, and then keep going back and forth and back and forth. And only by that means can it turn. So we thought, okay, let's first figure out how we're going to quantify this experimentally. So we put the cell on a micropipette, and this is a tracing of the flagella. Before we put any light on, we've averaged over many beats, and this is the kind of running average waveform through the beat. So the important thing about these waveforms is there's something called a power stroke, which is basically like a straight pivoting, and then a bent recovery stroke, and a power stroke and a recovery stroke. If the organism simply moved like an oar, stiff one way or another, it wouldn't get anywhere. That's a theorem known as the scallop theorem in low Reynolds number fluid mechanics. It has to push and retract and basically gets much more thrust from one motion than the other. It's a non-time reverse invariant motion. So if you then turn the light on, you will see a transient response where, in this case, the cis flagellum has vastly decreased the amplitude of its beating, and the trans slightly it's changed its beating. It's slightly more forward, but it's also smaller. So this imbalance between the two produces torques that lead to rotation. So we'd like to fundamentally build a theory on the basis of the torques associated with this imbalance. But again, here's the separation of timescales. If you actually imagine, say, 1.5 seconds of time, and you turn the light on at 0 and hold it there, the individual beats are at 55 cycles per second or something like that. So that's what that short time oscillation is representing. And the amplitude of the cis flagellum will decrease and then increase. The trans may change slightly in amplitude, but there's all this oscillation going on. We take the sum of these two torques, you'll get a net torque, which is oscillating with a running mean that goes down and comes up. And what we're going to do is ignore all those oscillations and do a kind of averaging over each beat to get a smooth, coarse-grained response like this central graph. And so, well, you have to quantify everything. You erect a little coordinate system. You keep track of when the beats begin and end by some little marker on the flagella that tells you when you've reached your maximum and minimum. And then you can actually calculate the force exerted by the flagellum and the torque around the center exerted by the flagellum from those waveforms using a calculational scheme called resistive force theory, which is the simplest approximation to the forces and torques associated with moving elongated objects. So it's an estimate, but it's pretty accurate. And the scale is piconewtons. And the scale of torques is piconewton microns, not Newton meters. But actually, a piconewton micron is a Newton. Something like that. Anyway, and you know, there are oscillating values of these through this power stroke and recovery stroke, but there are mean values, which are the ones we're interested in. And for instance, between cis and trans, there are ever so slight differences between the torques, which are responsible for this net turning even before a photoresponse. Okay, so when you do any measurement, we can convert what we measure experimentally with the beads into torques. And the theory that I'm going to describe is not built on the accuracy of this conversion. It's mainly as a sanity check for what's going on. So this is the adaptive response now expressed in terms of torques or the equivalent angular velocity of the object that would come by knowing an appropriate drag coefficient to balance the torques. So in this world of low Reynolds number, you don't have force equals mass times acceleration. You have force equals drag coefficient times velocity, sort of Aristotelian. So, and you know, there's some statistics on these time constants, but you see the same adaptive response. It's noisy, but it's there. Okay, so then you can look at the frequency response, just like we did with Volvox. And it turns out that that same kind of theory works where you have, this is not on a log scale, this is on a linear scale, you have this resonant response, and the data nicely conforms to it for particular values of those time constants. And actually, we can even get the phase shift between the response and the drive, just like you would in any kind of driven oscillator problem. Okay, so now excuse me while I make a slight digression to some math to show you from an applied math point of view how you would turn this into a proper theory. Right, so I presume you are all familiar with Euler angles as descriptions of objects in space, right? So I have an object which has an internal structure. I need 3 angles to describe its orientation. Say down from the pole, around this way, and then around its central axis. So in this problem, you take the usual Euler angle dynamics, where the angular velocities omega 1, omega 2, omega 3 around the body axis are related to the time derivatives of those Euler angles through relationships like this. But in our case, we have an omega 1 from the imbalance between the 2 flagella that's around this axis E1. We have an omega 3 from spinning around the central axis, but no omega 2 to a good approximation. So if you rewrite it as equations of motion for these angles, given the omegas, there it is. Okay. Now, the point is that there is a natural imbalance in the beating of the flagella that produces rotations around omega 1 and omega 3, and that's how you get a helix. We'll call that omega 1 hat. And then there's the photoresponse omega 1, which is the extra bit you get from the transient changes. And it's that photoresponse which is adaptive. That's the hypothesis here, that it, with these 2 time constants, is responding to the signal with a hidden variable. So we let that dynamics be adaptive, and then we're almost done. You just have to say, well, I know which way the eye spot is pointing if you tell me the Euler angles. I know the light intensity falling on that in terms of some Euler angles. And there's just a Heaviside function that tells you when the normal vector is facing the other way. And the only parameter of the problem is basically the strength, the maximum possible angular velocity you could get from those flagella. So instead of having to worry about the light intensity and how to convert that into frequency, we just say there exists a maximum omega 1 that you can get from the transient response, and we'll parameterize everything that way.
This probably doesn't matter, and it might be a bit technical, but does it matter if you actually put the Heaviside function in there, or if you put like the sort of cosine of the zenith angle in there?
So this function j has a cosine, has all the projections of the 2 vectors.
Oh, okay.
And then it's just a Heaviside of that.
I
So,
see.
Yeah, it would be, that would be a normal.
Because you don't want it working on the.
Yeah, so it's basically the cosine of,
Yeah,
it's the dot product of the 2 vectors.
great.
And then a Heaviside of that dot product.
That makes sense.
Right. Okay, and so you have only one relevant timescale in the problem that's universal, which is the angular frequency around the central axis, omega 3. You can scale everything with that. And then you end up with a 5-dimensional dynamical system. Okay, 3 Euler angles, which just evolve according to the intrinsic photoresponse, or the intrinsic rotation, and the photoresponse P, and then the 2 variables P and H, now called capital P and H, with dimensionless time constants, and we're all done. And then the signal is just given by some maximum value times that same projection, and there's the projection. So it's all there. Okay, so let's see if it works. So we just, for the moment, just start with some initial condition where we have the organism, in this case, facing away from the light, and we just let it go and see what happens. So remember, it's spinning around. The light is coming from the positive x direction, so it's going the wrong way. And what you'll see is you get these big dips in P as you get this transient response, and then you get a reverse response and transient reverse response, and that's what gives you the turning. And it dies away as the Euler angle tends to line up, and the projected light intensity is eventually going to 0. Okay, so when it lines up with the light, it no longer sees a signal. Which is not what I expected. I mean, sometimes I always thought that, you know, with an eye, it would line up so that the eye just faces the light all the time. And it doesn't. And then we realized that this is a confirmation of something that was predicted by some biologist, Schaller et al., many years ago, who again, like Jennings, without any math, realized that this was the only stable arrangement that could possibly exist for this to work. Because you see, their argument was it would be very hard for a biological system of this primitiveness to be precise about the intensity of light that it designed. But it'd be much easier to just say, if I get no signal, I know I'm right. Mm-hmm. So it turns out that that flagellum, which is dominant in the system to give you positive phototaxis, so if the light's coming from below, it swims toward the light, is such that the eye spot, when it moves on a helix, the eye spot is raked backward ever so slightly. So by the time it's actually swimming toward the light, the eye spot is in the dark. And any change away from that perfect alignment will suddenly bring the eye spot into the light, and it'll say, nope, that's wrong, go back. So it's as if the way I navigate toward you is by doing this, but it works. Because if I go either direction, I'll see you with this eye spot just on the equator. So it's pretty clever and non-trivial. Okay, now the thing you can do is a very simple calculation to understand quantitatively what's going on here. Imagine that I'm in the plane, and I'm just going to make a 90-degree turn to find the light. So let's say the light's coming this way, where I start this way. And so as I'm rotating initially, I'm going in and out of the light at the frequency omega-3. So I'm getting this periodic signal. And I have a dynamical system, and I have a response function called, which I call G, the Green's function, for the frequency response of the system. So my signal will depend on the orientation I have with respect to the light. And to first approximation, the Euler angle psi is just the spinning around the central axis, just goes like time in these rescale variables. So my signal looks like this. My response will just be some gain function associated with the response of the system at that frequency and some phase shift relative to the response, okay? And so this chi and this g are what I showed you before. We know those from the experiment. We know them in the theory as well. So if you just essentially integrate this forward, you get a very simple equation for the alignment with respect to the light. Phi t looks like 1 over a time scale times cosine phi. That's, like the equation of closing a door with a torque. And you can solve it. And this time scale, it's quite beautiful. It's the characteristic maximum rotation rate you can have in the system, divided by this response function with the cosine. And this inverse property there has this feature as a function of the product of the rotation frequency and the adaptive timescale. Remember, the G is a bandpass filter. 1 over G is 1 over a bandpass filter. So there's a strong minimum when you're tuned. This product being 1 means I respond, I adapt on the timescale of half an orbital period. And then if you move away, this timescale goes way up. So basically, if you're near this point, this minimum, then you will turn toward the light in the most rapid way possible. So I think this is like the summary of everything, that basically the inverse of the response function of this bandpass filter determines the timescale for motion. So you can, again, solve this problem just computationally. You can watch how the axis will line up with the light as the eye spot moves round and round, and eventually you find yourself with a fixed point pointing toward the light. But now we can track the 3D trajectories of turning cells and average over all of them, and we get this angle as a function of time, and it beautifully fits the prediction of the analysis with a timescale experimentally shown here, about a second. This is a box plot of the results, and the theory done 1 or 2 ways says exactly this from our measurements of the torques. So it kind of agrees. So now the denouement of this is this graph. I've now got 3 species spanning 3 orders of magnitude in number of cells, 2 orders of magnitude in radius, and I can plot for each of them, Chlamydomonas, Gonium, and Volvox, the product FR tau A that we got from these experiments. And if they are all about 1, that means that they're all tuned. And as you see, they are. So we can conclude that this tuning is an evolutionary conserved feature spanning this huge range of organisms that is allowing the simplest multicellular ones to function efficiently without any neuronal connections or any central nervous system at all. So there are many interesting issues here, especially about the robustness of this control strategy. In fact, if I go back to the previous curve, the fact that there is a minimum here is interesting, but it's a very broad minimum, which means that I don't have to be precisely tuned among my parameters to take advantage of this, but there is an optimization somewhere in the problem. And I think that combination of optimization and robustness to changes in the variables is an interesting lesson that at least we can see in this system.
And the number of solutions becomes smaller and smaller the closer your response or the TR and TA.
Exactly.
become.
So most of the time, statistically, we tend to be in about this region,
Yeah.
but every once in a while, any individual cell might be over here.
Yeah.
So it
But
becomes.
they have a smaller space. in
They have a
which
smaller
to-
space,
to
yeah.
work.
So that's kind of the story. But I would say that I think among the many other questions I've left open here, one of them is this idea that getting rid of a periodic signal is a kind of useful way of finding your way in the world. Seems to have been found by evolution multiple times. And I just wonder about what we could learn from that. Is it, as a control strategy, how broad is this in biology? And what biochemical structures to the chemical networks that make this happen allow it to happen? Next time I see you, maybe we'll have an answer. Thank you.
Thank you so much, Ray, for that really, really thought-provoking talk about some of the consequences of multicellular heresy. I think the next time I get in a swimming pool, it's going to be complete chaos. It's like thrash around.
Well, it's a different Reynolds number, so don't follow these rules.
Questions?
Well, that's absolutely fascinating stuff. Yeah, but
I knew there was a but.
There always is with this guy.
but it is about Volvocine green algae.
Yep.
And Volvocine green algae don't go anywhere. You know, they max out at Volvox. Correct.
Oh, you go anywhere from the point of view of a tree of evolution. Yeah,
Evolutionary sense.
of course.
So it's a good model organism, but or we can discuss that. And there are other systems that might work, and there are conoflagellates, which might be more attuned to thinking about animal.
Yes, and we've actually worked on them, and in fact,
and some of them do a Volvox kind of
yes.
thing. And have you worked on?
Yes. So we started working on them about 5 or 6 years ago, and there are a number of things. So conoflagellates are the closest living relatives of animals. So on the tree of life, if you look at the animal branch, it pretty much starts at sponges, and just to the right of that are conoflagellates. They're unicellular or small multicellular species that have a lot of features in common with what I've been telling you here, but they're directly related to animals.
And they're not phototactic.
Except for one that was recently discovered.
Oh, really?
Yeah. They can exist in the unicellular form. Some of them can transition between uni and multicellular depending on environmental cues. And there's one species called C. flexa that was discovered about 3 years ago in Nicole King's lab, which is a raft of cells held together by their collars. They're called conoflagellates because they have a collar of little polymeric structures that they use for filter feeding. And this raft is connected together by the tips of the collars. And that whole sheet can invert dynamically under changing light levels. And it swims very strongly in one configuration, not in another. And the net effect is it does phototaxis. Yeah, so we're actually finishing a paper on the mechanics and fluid dynamics of that very system. So I agree with you completely. Volvox is interesting, but it's important to go toward, I think, the more eukaryotic, animal-like species.
Or other plant-like systems as well. And there's an interesting counterpart, maybe the red algae and fungi don't have any flagella at all. And they're both been very successful at doing multicellularity. So there's that whole business of why things are becoming multicellular in the first instance, you know, and then the motors for making them work,
Yes.
the driving force. Really interesting.
Being ushered now.
You guys,
Okay, I think we have to finish.
we can swap places with them. Well, let's thank Ray again before we head outside and continue the conversation.